weekly element of our conference, and this is what we will introduce today. And you will have the chance to meet the chairs of all of these Life Science Tuesdays. In between of introducing the Life Science Tuesdays, you will also get a flavor of the virtual expo, where several companies, among them many of our sponsors, will introduce themselves on this online platform, and there will be possibilities to interact there as well. What is to be expected today is seen on the next slide. And I hope you can all see this slide. It documents the topics of the Life Science Tuesdays, which will be introduced today. And we will have 2 batches. So we'll first introduce, and this is what Helmut Berger will take care of, introduce the chairs of the first 6 Life Science Tuesdays, which are scheduled for 2020. Then we will have the introduction into the virtual expo. And then we will have the batch that I will introduce for the Life Science Tuesdays of 2021. So with these words, I would like to hand over to Helmut Bergler to start the introduction of the chairs for the first Life Science Tuesdays.
Thank you, Harald. I would also like to welcome you to this afternoon session. So now I will also share my screen with you. Just a second. Okay, so this is basically the same what Harald showed you, but only for those Life Science Tuesdays taking place in 2020. I would like to take this occasion also to thank the chairs for taking their duties here. I know this is, or we all know from the society, that this time it was much more complicated and much more work for the chairs also than a normal meeting. And we really appreciate your contributions and your help in setting up these meetings. Great thanks to all the chairs also, really. This was a great help for us all that we could get this running. So as Harald mentioned already, so there are 6 dates where each Tuesday afternoon a special topic is introduced, and the general format of each of these afternoon is that there are invited speakers, and there are also short talks and flash talks, and under certain for certain dates of the Life Science Tuesdays, there are additional activities planned, which will be shown to you by the chairs. The reason for this assembly now is that the chairs can present their own research. They will present, they give you a short overview of what the topic, and they will also mention the invited speaker. So I would now like to hand over to the chairs for the first Life Science Tuesdays, which is taking place in October already. And this is Martina Schweiger and Gernot Schabbauer. Martina, are you ready? And Gernot, are you ready for sharing your screen?
We are ready.
Okay.
I hope.
So thank you.
You have to finish.
Yes, I know. Yes.
Okay, so then I will share one of my screens. And full screen. So can you see it in full screen now?
Yes.
Yes.
Okay, super.
Can you hear me too?
I hear you. Okay.
Perfect. Hello.
Good. Hello. Good afternoon, ladies and gentlemen, colleagues and friends of biomolecules. It is our pleasure to introduce you to the first Life Science Tuesday of this ÖGMBT meeting with the topic Lipids and Metabolism. And the first Life Science Tuesday will take place on the 6th of October this year. My name, and it doesn't move on. My name is Martina Schweiger, and I'm one part of the duo chairing this session. And I'm assistant professor at the Institute of Molecular Biosciences in Graz. And I'm hosting the session Lipids and Metabolism, certainly because my own research focuses on lipid and metabolism. In brief, we are interested in adipose tissue metabolism, and my lab is mainly focusing on the plasticity of adipose tissue and how this plasticity affects whole body metabolism under hypermetabolic conditions like cancer-associated cachexia. We are investigating the communication between adipocytes and immune cells and neurons in adipose tissue and how a miscommunication between these cells contributes to disease development. And Gernot will now tell you what he is working on.
Yeah, hello from my side. So my name is Gernot Schabbauer. I'm associate professor at the Medical University for a couple of years now. And for even more than that, so more than a decade, I'm working on signaling, PI3 kinase and PTEN. Kept me busy for many, many years now. And over the years, I stumbled into macrophage biology. So actually, I'm an immunologist, and I was convinced by Martina to take part here. And recently, I also got interested in immunometabolism. So for some years, we are working now on arginine and arginase. And yeah, so in principle, that's it from my side. And I hand over back to Martina.
Thank you, Gernot. So then I can introduce you to our plenary lecture because we have the honor to listen to a presentation given by Andrei Kuda, who is head of the Laboratory of Metabolism of Bioactive Lipids at the Institute of Physiology of the Czech Academy of Sciences in Prague. So the focus of Dr. Kuda's work is the metabolism and regulatory actions of bioactive lipids, specifically related to glucose and lipid metabolism. Thereby, he is interested in a specific biomolecule deriving from de novo lipogenesis, the so-called FAFAs. So FAFAs are a branched fatty acid esters of hydroxy fatty acids. You can see this molecule here on the right side. And these molecules are specifically important for their anti-inflammatory actions, and they specifically affect lipid and glucose metabolism. And in his talk, Andrei Kuda will elaborate on these biological effects of this biomolecule of these FAFAs. And Gernot will let you know now about the other talks and science flashes we have.
Yes, so after Andrei's talk, then we will have the pleasure to hear a couple of short talks from researchers in Innsbruck and Graz, mostly about LAMTOR and how it affects brown adipose tissue homeostasis and adipose triglyceride lipase having a yet underappreciated function as a transacylase and the regulation of redox balance in lung cancer cells. Then we have a couple of innovative creative science flashes by young scientists from Graz and Maastricht about the inhibition of adipose triglyceride lipase and the role of ATGL in BCM, as well as the treatment of patients suffering from mitochondrial defects in neutral lipid storage disease. And then we will hear new aspects on the development of cancer-associated cachexia and how homocysteine levels affect atherosclerosis development. And now Martina will continue.
And we will finish the first Life Science Tuesday with a special presentation about life sciences on metabolism and aging, brought to you by Monika Oberer, who is representing the Biohealth Consortium of the University of Graz. And this workshop will give you insight into the research areas of the field of excellence, the biohealth, and also about DocFunds, the PhD training program to learn and explore life sciences. And we are very much looking forward seeing you at the first Life Science Tuesday on the 6th of October.
See you soon.
Thank you. Thank you very much, Martina and Gernot. Great presentation. So the next Life Science Tuesday is dedicated to RNA biology. And this one will take place at 20th of October. And it will be introduced to you by Brigitte Bertschi and Sebastian Falk. Sebastian Falk is from the Max Perutz Laboratory in Vienna, and Brigitte Bertschi is from the University in Graz. So Brigitte and Sebastian, may I hand over to you?
Yes, can you see my screen already?
Yes.
Okay, great. So it is a pleasure for me to introduce, so I'm Brigitte Bertschi, to introduce together with Sebastian Falk this second Life Science Tuesday on RNA biology. And first of all, we'd like to introduce ourselves. So I'm a group leader at the Institute of Molecular Biosciences at the University of Graz. And my research focuses mainly on ribosome biogenesis in eukaryotes and in the field of molecular and cell biology and biochemistry.
Yes, also a welcome from my side. Hello, my name is Sebastian Falk. I'm a rather new group leader at the Max Perutz Labs belonging to the University of Vienna. I started my team last year in March, so we are roughly 1.5 years old. And we are interested in nuclear RNA metabolism, RNA degradation, but also how small RNAs are made, so their biogenesis. And I'm a structural biologist by training, so we are using biochemistry and structural biology to address mechanistic questions of RNA metabolism in general. And we have the, the chance to host the RNA Life Science Tuesday. And as all of you know, RNA is a fascinating molecule because it's very versatile. It comes in different sizes and flavors. The ribosomal RNA, for example, is very huge, and small RNAs like microRNAs and piRNAs are very tiny with just between 20 and 30 nucleotides, but still they are very important. And connected to RNA are also a number of very interesting and important processes which many labs around the world are studying, starting from the biogenesis. So how are all these different molecules made? How are they degraded? And one of the really, really important topics also is how do these molecules, how does an RNA molecule know where to go in the cell? So what is the right destination? So therefore, we are very happy that we have two talks. The invited speaker, Marlene Oeffinger, will talk about RNA localization and transport, and Brigitte will tell you more about this. And the second highlight, which I will introduce later also a little bit, is the special presentation will be given by René Schröder, one of the most important figures in science in Austria, but especially in RNA biology, and she will give an historic perspective. So I will hand over to Brigitte, and she will tell you more about our plenary lecture.
So we are very happy. Yeah, we are very happy that we have secured the great speaker, Marlene Oeffinger, from the McGill University of Montreal. And she is working on different RNA maturation pathways in yeast, flies, and in human cells. And her main focus is on ribosomal RNA and on messenger RNA. She is actually originally from Austria and studied in Vienna. And then she moved to a famous RNA biology lab, David Tolavy's lab in Edinburgh, for a PhD thesis, and then went on to a famous nuclear pore specialized lab, Mike Rout's lab in New York, for a postdoc. And she has now her own research group in Canada where she combines these different aspects, so RNA and nuclear pores, and where she's interested in transport of RNAs from the nucleus into the cytoplasm. And there is a interesting question which is attracting researchers a lot, which is the question, do different RNA molecules or different transport substrates use different nuclear pores, and that's what her talk is going to deal with. So she will introduce finding that there is a special transport pathway using specialized pores for mRNAs. And the title is Nuclear mRNA Metabolism, Direct Specialization of the Nuclear Pore Complex in Yeast.
Okay, and as I already said, the second highlight will be a special presentation by René Schröder, which we will use to conclude the session and the day. And René Schröder also studied and obtained a PhD in Vienna. She then went for postdocs in Paris and New York, and then has been a group leader in Vienna at the Max Perutz lab, which is formerly known as the MFPL to most of you. I mean, she received the Wittgenstein Prize and the Eduard Buchner Prize, so really prestigious prizes that you can obtain. And she and her team have worked on many, many different aspects of RNA biology, starting with catalytic RNA ribozymes and the interaction with antibiotics. How do RNAs fold, and how is this folding facilitated by proteins that are called RNA chaperones? Her lab made important contributions to RNA interference. They worked on transcription and its regulation, and also very creatively, René's lab always used new technologies that came up by using aptamers and SELEX. So we're really, really happy that René agreed to give a special presentation about giving a personal historical perspective about RNA research. And I think this will be interesting to every student and researcher to see. So we are really, really happy.
So let me just finally show you the schedule of our Life Science Tuesday. So we will start out with Marlene's talk on mRNA metabolism and connections to the nuclear pores. And then after discussions, we will have 3 short talks and followed by the science flashes, and all of them cover different aspects of RNA biology using different types of RNA molecules. And after subsequent discussions and virtual expo, we will finalize our session with René Schröder's talk. And of course, we are looking forward a lot to seeing you on our Life Science Tuesday on October the 20th. Thanks.
Thank you. See you all.
Thank you very much, Brigitte and Sebastian, for the great presentations, and also for setting up this interesting program. So the next Life Science Tuesday is dedicated, or is called Structure to Function, and is chaired by Tobias Madl from the Medical University in Graz, and by Elif Karagöz from Max Perutz laboratories. Elif and Tobias, are you ready to start?
Yes.
Okay, thank you.
I'm trying to unmute myself and it wasn't successful. Hello, everyone. I would just like to share my screen right now. And hi. I'm Elif Karagöz. As Helmut mentioned, Tobias and I are very happy to chair the session of Life Science Tuesday Structure to Function, which will be taking place in 3rd of November. We would like to also briefly introduce ourselves. I'm Elif Karagöz. I am a group leader, a fresh group leader at Max Perutz Laboratories in Vienna. And in my lab, we aim to provide mechanistic understanding to protein folding and stress signaling pathways. And to do so, we combine structural approaches with biochemistry and cell biology to dissect these pathways from atomic to cellular level. And Tobias, stage is yours.
Yeah, thanks a lot. Welcome from Graz, from the Medical University. I'm associate professor here, mostly working in integrative structural biology, using a lot of solution techniques such as NMR, spectroscopy, SAX, and sometimes also X-ray crystallography and other techniques. And we are particularly interested in how intrinsically these sort of proteins are involved in signaling, in particular in pathways such as Wnt signaling, FOXO and p53 signaling. Yeah, and we love to use integrative structural biology to answer questions and related to interaction networks and yeah, how modifications and distortions in these protein networks are associated with diseases. We also have a small spin-off that we just started recently, where we also would like to follow up yeah, our basic research a bit more in respect to drug discovery and development.
And we would like to introduce you further to our Life Science Tuesday. As you know, is that the theme of our session is structure to function. We focus on this theme not only because it falls into our research interests, but mainly because the use of integrative structural methods have been revolutionizing the structural biology and cell biology field in recent years. When we talk about integrative structural biology, we talk about basically combining data from various methodological approaches using NMR spectroscopy, crystallography, to bring information from various spatial and time resolution to understand the function of macromolecules and how they work in high precision in the cellular complexity. And we would like to give you some examples of how integrative structural methods basically provided mechanistic understanding to functional biological processes. One of them is actually the work of Tobias's and my collaborative efforts on solving the structure of HSP90 chaperone with one of its clients, tau. HSP90 forms very dynamic complexes with its clients, and we use state-of-the-art NMR approaches with small angle X-ray scattering to figure out how this protein recognizes its substrate molecules, especially this unusual client tau. And basically our work together revealed the substrate recognition principle of this highly conserved chaperone that remained elusive for decades. And with that, we would like to continue. Tobias, please.
Yeah, that's already the bridge to our keynote speaker, Teresa Calomagno. Teresa has really, yeah, she has really solved some quite challenging large protein complexes using an integration of different techniques, and that's one of her recent examples, the BOX-CD enzyme, which methylates ribosomal RNA at the 2' ribose. And there she really had to combine yeah, different techniques, structures of the individual domains obtained by X-ray crystallography, overall orientation of the proteins, subunits within the complex using X-ray and neutron scattering and NMR information, defining the positioning of these individual domains. So that's really, I think, a very nice example on how you can combine yeah, the best of every technique to solve the structure of a very large protein, multi-protein RNA complex, with the aim, of course, to understand the underlying biology that will then help basic research and also promote and set the base for drug discovery, for example. Teresa studied in Naples, chemistry, and then moved to Frankfurt for a postdoc, and later on for a postdoc at Scripps. And in 2001, she moved back to Germany, where she started her own group in Göttingen, and then was very successful moving over to the MBL in Heidelberg. And since 2015, she's now full professor in Hanover and also at the Helmholtz Center in Braunschweig. She is very active not only in structural biology, where she studies structure and function of ribonucleoprotein complexes, but also in NMR method development, because if you are working in integrative structural biology, you often hit the limits, and then you have to find new ways to overcome these limits. And yeah, that's where she has been very active also in method development to study large complexes using novel, innovative solid-state NMR approaches, for example. Her work has also been recognized by several awards, and she was also organizing large conferences in the field, the ENC, the EMO conference. So that's quite a well-known conference in the field. So we're very much looking forward to a plenary, and we are now accepting abstracts until the 26th of September. So we're very much looking forward to receiving your abstracts and seeing you on the Life Science Tuesday, 3rd of November.
Thank you very much.
Thank you very much, Elif and Tobias. Great presentation. The next topic is organelles and transport. And this session is chaired by Anita Emmersdorfer-Augustin from the Technical University in Graz and by David Theis from the Medical University in Innsbruck. So Anita and David, looking forward to your presentation.
Hello, Helmut. Can you see our screen? No?
Yes, now we can see the screen and we can also hear you.
Can you hear me? Excellent. So, first of all, I would like to thank Helmut Berger and also Harald Pichler and the entire ÖGMBT organizing committee for coming up with these excellent ideas of Life Science Tuesdays. I think this is a quite good idea to present research going on abroad and that is related to Austrian sciences, to the Austrian life science community. My name is David Theis, and I'm sharing this session together with Anita Emmersdorfer-Augustin. Our keynote speaker is Hesso Farhan from the University of Oslo, and on November 17th, he will present in our session Organelles and Transport, a talk about the regulation of ER proteostasis by mechanobiology. Basically, this is at the heart of cell biology because it integrates the cellular architecture, how cells are built, with external information that cells receive from their environment. And this is exactly what cell biology, the biogenesis of organelles, and the transport of molecules within cells is all about. Maintaining a dynamic network of membranes, organelles, and biological functions that is able to respond and adjust to intra and extracellular stimuli. And also to share some bit of news, Hesso is currently a professor at the University of Oslo.
Your microphone just muted.
David, you have to switch on your microphone. David? David?
Yes, can you hear me? I don't know.
You muted your microphone.
I did not. Oh, where is it? No, it should be active. Don't you hear me?
No, it's okay. No, it's okay.
You just the presentation mode is stopped. It's your whole desktop.
Okay, that's good.
Maybe just stop the screen share and restart it again. Is PowerPoint open?
PowerPoint is open.
Perfect.
Can you hear me now?
Again. Yes, we can hear you. Starting slide.
Maybe it's the connection to the West, who knows? All right, so Hesso is currently professor at Oslo, but we successfully recruited Hesso to the Biocenter in Innsbruck, to this beautiful building, and we are quite pleased that he will join us in Innsbruck starting with January as a professor of physiology in the Biocenter and at the Medical University. Another news that I would like to share with you, especially targeted to people who just finished their master studies, is that we have been successful in establishing a new PhD program, Cellular Basis of Diseases, which is an interdisciplinary PhD program on the molecular control of metabolism and inflammation. And we have 15 PhD positions available in that program. And you can still sign up and send your application to us until the end of this month. And you can use that webpage to sign up for this PhD program. It's an FWF-funded Doc Funds PhD program. And with that, I will hand over to Anita, who will explain to you the session that we have planned. Anita, maybe you can say a few words.
Thanks, David, for handing over. I hope everybody can hear me just fine. A couple of words about myself. I'm a rather new PI. I just started a young researcher group at Graz University of Technology, funded by Biotech Med Graz. And we are investigating how lipid perturbations and modifications influence diverse cellular processes. For example, stress response pathways, but also sorting mechanisms and transport phenomena. So this is actually the angle I am coming from and how I ended up in this session. Well, to keep the story short, we are still urgently looking for abstract submissions for our session, Organelles and Transport. So the abstract submission deadline has been extended until October 10th. And we are really addressing rather young researchers, so master and PhD students, but also postdocs. So dear PIs, please pitch the session and advertise it in your groups. It's really important to spread the word and show the results of your research to the research community to get some feedback. So we are looking for short talks and science flash presentations. And also you should never forget about the fact that there will be talk and poster awards. I know a lot of people who won one of those. They're great for your reputation, that they're also going to bring some money. So I'm looking forward to seeing most of you again during our session. And well, yeah, thanks for listening.
Thanks again, and looking forward to meeting you on November 17th. Bye-bye.
Bye-bye.
Thank you for the presentation, David and Anita. Looking also forward to your presentations over there. So the next topic is microbiome innovations. And these sessions will be chaired by Christine Meusel-Eichinger and Gabriele Berg. So they are well-known experts in the microbiome field. Unfortunately, both of them are on travel, and so they did send me an file with their presentation, and I will open it and hope it will run smoothly. Okay, so please tell me whether you can hear it. Is the sound okay?
Not hearing or seeing anything except you.
Okay.
Share screen and computer audio at the bottom.
Sorry. Do you see it now?
No, we only see you video.
Okay. So you see more than me, actually. Just a second.
Really looking forward to our session. I'm chairing.
Is it okay now?
Now we can hear and see and hear it.
Okay. I will start again.
Hello, everybody. I'm really happy to be here today with you.
The sound okay?
I'm here to pitch for
Yes, it's fine.
Tuesday, which is on microbiome innovations and will be held on December 1st. So I'm really looking forward to our session. I'm chairing the session. My name is Christine Meusel-Eichinger. I'm working at the Medical University of Graz, and my co-chair is Gabriele Berg, who is working at the TU Graz. We are both professors for microbiology, in particular microbiome research. So we are particularly interested in the interaction of the good microbes with the plants and the animals, and of course, particularly the humans. So of course, you know that microbiology, microbiome has received a certain boost over the last 10 years. and microbiome research has become a very important field in the field of microbiology with respect to diagnostics, understanding certain diseases, but also understanding the beneficial potential of certain microorganisms. Gabi and me, we are active also on YouTube, so if you're interested to learn more about these topics, please visit us on YouTube, on Twitter, or on our websites at our universities. Actually, we're also here today to represent Amici, who is the Austrian Microbiome Initiative. And so you see in Austria there's a lot of microbiome people, and we are always searching new experts and new followers for this specific group. On December 1st, of course, we have also a fantastic speaker. I'm really happy to have her on board. The speaker of our session is Professor Gillian Patterson from the University of Vienna. And you might know her already. She is a recent ERC recipient, and she's very interested in the interaction of certain microbes and clamps. And she will be talking about how environments shape microbiome innovations in the natural world. I'm really looking forward to your talk, Gillian. It will be fantastic to listen to you. If you're working in the field of microbiome, microbiology, please don't forget to submit your abstract for short talks and science flashes. The deadline is already very soon, so it's October 1st. Please send your abstracts. We're looking forward to receiving them and to inviting you to present your work. That's basically it. Thank you very much for listening. Please join us on December 1st. I'm really looking forward, and thank you for your attention.
So there is still some time to send your abstract for the microbiome session. So the last session this year will be chaired by Rolf Breinbauer from the Technical University and Christian Becker from the University in Vienna. And the topic is chemical biology. And Rolf and Christian, looking forward to your presentation.
Thank you very much, Helmut. We are very glad that for the first time at the ÖGMBT meeting, we will have a session about chemical biology. And I feel very proud that Christian Beck and I have been selected to chair this session. It will take place in December, as already mentioned. And the field of chemical biology is something that has established itself quite recently. And we are very proud that we could recruit Stefan Sieber from the Technische Universität in München as a plenary speaker for our session. And I will introduce Stefan to you finally, sort of in the last part of our presentation, but want to head over to Christian Becker to define the field of chemical biology, because it might be new to some of the people in the audience.
All right, thank you very much, Rolf, and welcome all. It's a pleasure to give a definition of chemical biology, which is not so easy because it's a very dynamic and quickly developing field, already 2 decades old, I would say, and it differs from what we usually call biochemistry by not looking really at the chemistry of biomolecules, but rather by using creative chemistry to somehow elucidate biological problems, to solve biological questions we have, or to manipulate more complex systems by using chemistry, analytical tools, and so on. And its dynamic development and its, let's say, already quite big name in science is also reflected by the number of journals that are using the name chemical biology. And here's just a small number of them, probably the ones that are most famous in the field. And if you look around, many departments, research institutes, research initiatives, and so on have started to use the name chemical biology over the past 10, 15 years. And the name has become a household item in it's the interface of chemistry and biology. And in our session, we would like to cover I guess it will be possible to cover all of chemical biology, but at least offer the opportunity to cover a large part of this. And therefore, I will define what at least Rolf and I understand of being part of chemical biology. And the first of all are bioconjugation and bioorthogonal reactions that you need to manipulate biomolecules or other molecules in complex environments such as cells and organisms. And you need them to introduce dyes, tags, probes, being it for structural investigations, for tracking molecules in cells or organisms. And at the same time, you also need libraries, combinatorial libraries of molecules to actually elucidate the desired biological responses or to actually interfere with specific biological systems. And below these kind of headlines that I've put in here for chemical biology, there are many other topics that come into play, and the selection that we show here is not complete and of course biased by our personal interest of research topics. And there is peptide protein synthesis, there's glycobiology, there are specific classes of enzymes, overall modifying enzymes, the interest in understanding patterns of post-translational modifications in cells, of investigating or introducing connections between molecules that are typically not present in biological systems by using molecular glues and other systems. And to do so, of course, you need a variety of approaches, evolutionary approaches. You need proteomics to interrogate the system. And that topic will come up when Rolf will introduce our key speaker of our Life Science Tuesday. And of course, there's nucleic acids and aptamers, which are also covered in other sessions in this meeting. So with all of these keywords, we hope to motivate many of you to submit abstracts for chemical biology, and let's have a great Life Science Tuesday in December. And with this, I hand the word back to Rolf for saying a few more words about our speaker.
Thank you very much, Christian. And so you see that chemical biology is a field which brings really chemists and biologists together, and some of the most prominent representatives of the field combine both expertises in the very same lab. And Stefan Sieber is one of these outstanding scientists. He studied chemistry in Marburg, was a PhD student in Mohammed Marahil and Chris Walsh's lab in Harvard, was then a postdoc in Ben Kravitz's lab at Scripps Research Institute, and is now sort of chair of organic chemistry at WTO in München. He has received twice the ASE grant, and also most recently he received the €1 million Future Insight Prize from Merck. And the title of his talk is Chemical Proteome Mining to Fight Multi-Resistant Bacteria. Stefan uses activity-based protein profiling, identifying new targets, which might give us sort of a new twist in handling one of the most pressing and urgent medical needs which we have to our days, and which become even more relevant in the future. So we are very excited to have Stefan as an invited speaker in our session. And finally, we want just sort of to inform about the deadlines. The abstract submission deadline is the 15th of October. And so we want to invite you to send contributions. We will select the short talks and the science flashes. And yeah, we would love to have you in December 15 on our conference. Thank you very much.
Thank you. See you there.
Thank you, Christian, and thank you, Rolf, for the great presentation also. Generally, I want to thank all the chairs from the first part here, and it was really impressive what you showed here, and we are really looking forward to the Life Science Tuesdays. Now all the participants have the chance to talk to the individual chairs. So when you want to in breakout rooms, so when you want to talk to the chairs, just write the number of the chair of the Life Science Tuesday. So this means 1 and a minus, or 2 and a minus, and 3 and so on, depending what Life Science Tuesday chair you want to talk about now, for in front of your name, and then you will get sorted in breakout rooms, and you can discuss with your chair for I think 10 minutes, and afterwards Andrea will give you a guide into the how to enter the virtual expo. So Walter, are you ready for sorting the people into the
I'm ready.
rooms?
So please just rename yourself. So you can do this by going over your video in the view. You have 3 blue dots on the upper right and click rename. And what you do is please the Life Science Tuesday session you want to join. So add the start with a number like 1_2_3_. So this is now only, by the way, for the first 6 Life Science Tuesday. So the breakout rooms for the second part will be a separate breakout room series. Chairs, please also write their number in front.
And as an update, in about 10 minutes, we will all be back automatically in this main room and then go on with this virtual expo. And afterwards, the second batch of Life Science Tuesdays will be introduced.
Harald, would you please maybe share this the slide with the Life Science Tuesday so that people can see to which?
Yeah. Yes, of course.
Thank you.
I forgot. Sorry.
A short request to Rolf Breinbauer, if he can hear me, to put the number of his Life Science Tuesday next to his name. Thank you. That's Life Science Tuesday 6 you're chairing. Thank you.
Please put the numbers in front because at the back I can't see them. Unfortunately, the drop-down box doesn't show the end. Okay, so has everybody changed their names to the breakout rooms they're interested in? So if anybody later decides it, I can still add people, but yeah, you can always add the names. Okay, then if those other people who are interested have changed it, I'll open the rooms now. How long did we say? 15 minutes? 10 minutes? Okay.
I think 10 minutes.
Mm-hmm. Okay, you will be automatically moved into the breakout rooms and you have 10 minutes time. Afterwards, you will be all automatically set back. People who haven't set anything will stay in the main room.
One important information before maybe when you enter the room, then switch on your video and your unmute your microphone, please.
Now I know what we forgot. Some elevator music for the people who stay in the main room.
maybe in the meantime, as we have a lot of exhibitors here, exhibitor representatives, may I ask you add to your name also the company you're coming from? So that might be good for later. And also for other participants, please add to your name also your affiliation by renaming with the 3 dots on the right upper side on your image.
Alexandra, maybe all those who are waiting to be introduced as virtual expo participants can just put expo in front of their name so we can show them easier.
I don't think that it's necessary, because when the people come in, when I start the presentation, I will ask them to show the videos, so to turn off the videos, so they should be the only one who will be present. And
okay,
thanks.
Sorry, Alexandra, may I ask 2 questions? Please, maybe I did something, but one thing is, how do we get into the breakout rooms? And also, how can the visitors see the exhibition booths?
Yeah, to the breakout rooms. So the breakout rooms are managed by Walter. So if they are planned breakout rooms, you have to, for example, like now, to show your interest by renaming, or some breakout rooms are also done by randomly. So if you want an unplanned breakout room, you have to inform us about this so we can arrange it. And the second question was how the participants can visit the virtual expo?
Yes, but just the breakout rooms. What breakout rooms do we have?
Now the breakout, it's dependent on the session we are in. So now the breakout rooms are for the Life Science Tuesday chairs. For example, at the Life Science Tuesday, there will be breakout rooms. They're called mixer, so just randomly. And there will be also breakout rooms for the science flashes. Okay, so they are dedicated to, yeah.
For today?
No, during the Life Science Tuesdays.
I see, only, so not for today.
Yes, for today we have this breakout room now, and also at the end of the second part of the presentations of the Life Science Tuesday chairs. Yeah.
So now if you want to go to a breakout, you can go to the breakouts for Life Science Tuesdays 1 to 6.
Yes.
Yeah. Just put the number in front of it. I'm monitoring and will automatically assign you to the correct room. Or if you want to see 2, just do first number, go in, then leave the room, add second number in front, and I'll assign you to. So we basically have 2 ways of doing it. So we basically have technically 2 possibilities, is basically to split people up by random. Let's say 10 people per rooms, and we say we want 3 or 5 rooms, something like that. And the second one is a manual assignment like now. So basically people say, in which room do I want to go, by giving the number in front of the room, and then I assign them by
hand. Michael Bergmann, do you maybe want now to make the technical check for your presentation?
Yes, fine.
Yes?
So I just I put my video.
I don't think so.
Do I?
Ich sehe nichts auf der Liste.
Ich habe sie da oben auf unserer hier.
Okay, presentation view. Great. Okay. Yes.
So
great, it's working. Yes.
Okay. So I put stop. Wait a minute. Stoppen.
Yes. Great.
Awesome, yeah. Thank you for testing.
Yes, habe ich
Are there any other questions or anything?
We don't have to show a presentation, right? We just introduce ourselves quite quickly and
no, you just say
it.
hello. Just I will ask you to show you, and I will thank you, and you just say hello so the people can see you. You see your faces and your names, and then we will make a virtual tour to the virtual expo.
Ah, cool. Okay.
Okay.
Thank you.
I have a question. If someone of the exhibitors who are here now, is someone now online or available for a video chat within the virtual expo?
I am available. I'm Carolina from Ouroboros Instrument.
From Ouroboros, okay. Then I just want to check whether I see it.
Yes. Should I put in the booth the active video? Yes.
If you're logged in the booth, and yes, I see
I'm
it.
already.
Great. Great, thank you. So I can just show it how it works, and that's good if we have someone who is also online.
But the strange thing is we didn't sign up for the video chat. We just took the basic booth, but for some reason I can also activate video chat in my chat room.
Okay.
That's strange.
So you have luck. I will look afterwards. It's switched off, maybe. No, it's working. It's okay.
I didn't
just
plan to use
okay.
it, but I was a little bit surprised. No.
Okay. So if it's working, so just use it.
Okay.
Do you want me to keep the booth with the video chat activated?
Yes, please. It would be great, because I will make a virtual tour afterwards, and it is good if some booths are active, and so I can show how it looks like when a booth is active on text or video chat. Thank you.
Perfect. Thank you.
Hi, Alex.
Hello.
We are also active, but on my computer the video is not working because I have some problems with the camera. But
okay.
But I can chat. I tested it out with a few colleagues already.
Okay, great. Thank you. So, Walter, how many minutes do we have left after the other participants come back to the main room? So I think Julia Feichtinger want also to be added to Life Science Tuesday 2 breakout room. Is that right?
Yes, please.
Yes, Walter, could you please?
Frau Kassidov?
Ja.
Ja, Bergmann Andreas von Savos Kandig ist hier. Grüß Gott.
Grüß Gott, hallo.
Ich sehe gerade, ich habe einen schrecklich gefärbten Hintergrund.
Wird an der Kamera liegen wahrscheinlich.
Wird an der Kamera liegen, ja. Also viel besser wird es nicht. Also unser Stand ist besetzt. Anchatten kann man uns.
Sehr gut.
Videomäßig können wir gern so bleiben, aber wie gesagt, besser, glaube ich, wird das von der Einstellung nicht.
Du wirkst von der Bewegung her wie ein Roboter.
Danke, das ist
langsam, bitte, Adi.
Sehr freundlich von dir.
Du bist kein Bot. Schaut's im Hintergrund meine Flaschensammlung? Corona-like?
Oh.
Mhm.
The breakout rooms are now closed and everybody should be back again.
Okay.
Okay, so we will switch now to English again.
Mhm.
So, Helmut, Harald, are you here?
Yes, we are ready.
I'm here, and I'll share the screen. So,
I think Alexandra will.
Yeah, Alexandra will do. Yeah.
Okay, thank you. So welcome also from my side. I'm very happy to cut the ribbons for our popular exhibition. As the whole meeting is in a virtual format this year, I'm also very excited to introduce you to the virtual expo, which will be also longer available than you're used to, so more than 2 or 3 days. The virtual expo will accompany us throughout all Life Science Tuesdays, so the next 6 months. Why do we have this? Because every one of you knows the Ökumenical meetings are also not just for the scientific part, but it's also accompanied by a very big exhibition. And we appreciate our exhibitions not only because of the competent first-hand information about the products and services, which are essential for our all-day work of scientists, and we do also not only appreciate it because of the useful and funny giveaways we usually get at the booth, but also because of the representatives themselves. So many of them are also here in this Zoom room today. I would like to welcome there and ask them also to unmute their microphones and to show them so we can say hello. And
hello.
Hello Matthias.
Hi there.
Hello Chantal. Yeah.
Hello, good afternoon.
Hello. So we have a lot of companies represented here today, and which are also represented at our virtual expo. But before giving you the facts and to make a virtual tour with you, I would like to say also thank you to all of you for being with us, for supporting us, and you have proven loyalty and innovative spirit that you are following us to this innovative format. So huge thanks from all of us.
Welcome.
You're welcome. Thank you for you.
So and now I will share my screen to make a virtual tour with you. Just give me a second. So now you should see the ÖGMBT webpage of the annual meeting. So how we can visit the virtual expo, it's very easy. It's available for everyone, not just for registered participants, but for everyone who visits our webpage. When you go to Jahrestagung and Virtual Expo, you are here at this page. And actually, so I'm now in the detail view, I will switch back. Then you will see the overview page of all our exhibitors. You can see it, some exhibitors have this sign here at the bottom, which means that they are available for online chat. Some of the exhibitors have also this video icon, which means that they are also available for a video call. So it was very important for us to keep this interactivity during the whole meeting. And so the virtual expo is not just a platform where the exhibitors can present their portfolio, but they are also available for interactions with you for text chat or for a video call. So let us visit maybe 1 or 2 exhibitors. So we have here, for example, yeah, the exhibitor MP Biomedicals. So
Thanks,
it's
Alexandra. Thanks to show our booth first.
Yeah, so I invite everyone to visit every booth, of course, but I just picked one to see what we can look for. So the exhibition is available 24/7. You have here several mediums. So there are some videos, sometimes. Always, not sometimes, always very informative and sometimes also funny videos. And you have here the information of the company. Also, for example, in this case, MP Biomedicals is also presenting a webinar at One Life Science Tuesday. You have your contact details, and you have here different kind of informations, of documents for products. Yes, what is important at the what you can do also is you can get in contact. For example, you can see here text chat available. So by clicking this button, you are asked for your name and email address, and you can get in contact with the exhibitor. This button is only available when the exhibitor is online, so you can be sure that you will be answered shortly. Then you have a function if you want to talk to the exhibitor one-to-one to a specific, to make a specific appointment. You can click here for this calendar icon, and you can choose one of the possible dates which the exhibitors offer, and so you can schedule a one-to-one video call with the exhibitor. Yes. Returning back, so you can also, for example, see here if the exhibitor is available for a video chat. You can also click this button. It's the same button for text chat and video chat, and you can get in contact with them. So yeah, what is also very important to know for all of you who knows the ÖGMBT annual meetings, we also run, again, also in this virtual format, we will run an exhibitor quiz. So all registered participants have the opportunity to win prizes, but this time also not just one prize, because we run the exhibitor quiz at every Life Science Tuesday. So at every Life Science Tuesday, you have the chance to win prizes. This can be vouchers for trips or vacations or other surprises by the exhibitors. And yeah, how it's working. So you just visit one exhibition, one booth, one exhibitor booth. You look for the question. The questions are not online now because it will start at Life Science Tuesday. But so when you look at 6th of October, there will be questions present at the booth. So you look for the question and try to find the answer by clicking through the information. But if you also cannot answer it by yourself, you can contact the exhibitor. And so you have actually to take part, contact the exhibitor with a text chat or a video call to give your answer. So if you don't know the answer, I'm sure the exhibitor will help you to find the right answer. And the exhibitors will collect your name and your email, and those of you who will visit at one Life Science Tuesday, the most exhibition booths, they will win a prize. Yeah, it will be then sent via surface mail. So it's worth it to visit because of the information, because of the interaction with the exhibitors, and also you can win prizes. So if I've forgotten something very important, please let me know. But I think this was the most important. Were the most important part. Yeah, and now I would like to hand over to Harald for the scientific part.
Thank you, Alexandra, for this overview. And now it's time to go for the second batch of introductions of the Life Science Tuesdays. And with this, I share this slide. So we have those Life Science Tuesdays that are to take place in, sorry, that was the wrong slide, the wrong direction, that take place in 2021. And for organizational reason, I do something one should never do in such a format. I would ask Life Science Tuesday number 10, so omics data analysis and integration, to present first, because one of the chairs needs to leave this meeting in a few minutes. And this number 10, Life Science Tuesday, is presented by Julia Feichtinger and Thomas Rattay. Please share your information with us.
So I hope you can see the slides. Welcome from, welcome from our side. We're very happy actually to introduce the Life Science Tuesday number 10 about omics data analysis and integration. This is a bioinformatics topic, and this Life Science Tuesday is also very much supported by the Austrian bioinformatics community, the Austrian bioinformatics platform, kind of as a co-society behind the ÖGMBT. And we have selected among the many topics that are very interesting actually in respect to bioinformatics, the omics data analysis and integration as a topic for an individual session. There will be a lot of bioinformatics, of course, also in other sessions, and you will definitely have the opportunity to learn a lot and to discuss a lot about the many ongoing directions in our field. First, I would like to introduce the chairs and would like to start introducing Julia Feichtinger. Julia works at the Medical University of Graz. She initially did her master's and her PhD in the UK. She studied bioinformatics and did a PhD in cancer studies. Then went back to Graz and did a postdoc at the ADSIP in Graz, and further became PI at the Gottfried Schatz Research Center for Cell Signaling, Metabolism, and Aging. Her work covers a couple of different areas, as very typical actually for bioinformaticians, that we are not only running in one direction, but typically have a lot of interfaces in our network. And so her publications cover aspects of cell biology, aspects of biomarkers, omics studies, and everywhere there's a lot of data analysis and bioinformatics involved. And so I hand over to Julia now.
Yes, thank you. So it's my pleasure now to introduce you to Thomas Rattay from the University of Vienna. He's a full professor and he's heading CUBE, the division of computational system biology. And his research focuses on genome and metagenome analysis, as well as on system biology and computational genomics. And he really has a lot of expertise in developing and applying computational methods to interpret large-scale sequence information. So it's really a pleasure to co-chair this session with him. And with that, I hand over to you, Thomas, and you will now introduce our key speaker to you.
Yeah, thanks very much, Julia. Our Life Science Tuesday is a bit ahead on 23rd of February, but we're very happy that our keynote speaker has agreed almost immediately when we asked him, because we think that Oliver Stegle from the German Research Center, Cancer Research Center, ideally represents the research that is typical basically for a bioinformatician who is involved with a lot of omics data analysis, integration, visualization. His particular area are computational genomics and system genetics. These are a couple of keywords, but it all boils down to the super important and kind of central question of the relation of genotypes and phenotypes. We all know this. He particularly works on spatial heterogeneity. We all know that the cells are, of course, separated by the spatial position. He works with cell-to-cell heterogeneity, because even in the same locus or in the same kind of spot of an organism, the cells are not doing exactly the same. He works on genetic heterogeneity and the role that genetic heterogeneity plays actually for the phenotypic heterogeneity. And all these questions actually he approaches from the kind of data point of view, which means this group is involved in a lot of different projects where a lot of data are generated first, of course hypothesis driven, but still with the view actually on the entire system and with the kind of unbiased omics kind of data generation. Here you see 3 examples: genome, metabolome, proteome. They have more techniques that they are using, and then kind of a whole battery actually of tools and approaches are needed to bring all this data together and to derive interesting scientific results. It all starts kind of with data reduction. Here a lot of bioinformatics is involved. Then typically a lot of statistics, specialized statistics are necessary. In particular, if heterogeneous data from different omics methods needs to be integrated, that is definitely not trivial. It's not just running some simple statistic test. There's a lot of effort for normalization and for testing and for combining the data sets required. It of course needs sophisticated visualization to understand the data, to in the end being able to interpret these data in a clever way and in an interesting way. And for all these areas actually, Oliver represents or brings the expertise. He represents basically a person who's experienced with all these kind of aspects, and so we are very, very happy that he will present his previous and of course also ongoing research in future directions actually in his keynote session. With that I hand over back to Julia now.
So after this exciting keynote, we will have short talks but also science flashes. So please submit abstracts so you can contribute to this exciting session. And as you can see here on this slide, this session is all about the analysis of any kind of omics data. And this includes also the development of new algorithms and tools, and topics on omics data integration are particularly welcome. So if you want to contribute, the deadline is on the homepage. It's actually the 23rd of December. And finally, I really want to wrap it up by inviting you to join our Life Science Tuesday on omics, data analysis and integration. And don't forget, it takes place on the 23rd of February. And we're looking forward to this session and to everybody who wants to listen in. And and to hear all the excited topics, the excited keynote, short talks, and science flashes. So with that, thank you, and well, see you then.
Sorry, I was muted. Thank you, Julia. Thank you, Thomas, for presenting your Life Science Tuesday of February 23rd. And now we revert to the original order of the Life Science Tuesdays and come to Life Science Tuesday number 7, which is presented by Georg Klima and Klaus Graumann and will take place on January 12th.
Okay. Can you see the slide?
Yes, that's fine.
Yeah, Klaus, I think we can see it well.
Just the presentation.
Very warm welcome also from my side.
Doesn't work. Sorry for that.
Try clicking on presentation mode a second time. Sometimes
do it already the 5th or 6th time.
Okay. Do you see your presentation in?
I only see the presentation now.
Okay, maybe stop the share and restart PowerPoint.
Okay. Yeah, obviously it's frozen. Sorry about that.
Ah, okay, that explains it.
Maybe we should revert the order once again until I fix that, because it seems I have to restart again. Can we do that?
Yeah, we can do that. So we'll come back to you in about 5-6 minutes.
Yeah, sorry for that.
Okay, no problem. This is what this My Life Science Tuesday session was also planned for, because we get to know these technologies, all of us. Okay, so Life Science Tuesday number 8 will take place on January 26 and will be presented and chaired by Stefan Schild and Sabine Kinesberger. Looking forward to your introduction.
So, good afternoon, everyone. First of all, I'd like to thank the organizers for the great idea to put together these Life Science Tuesdays. So, Stefan and myself, Sabine Kinesberger, we are really excited to chair this session on infection and immunity on January 26th. We are both affiliated with the Infection Biology Unit at the Institute of Molecular Biosciences in Graz. And our research focus on the elucidation of molecular mechanisms of bacterial infections. Our model organisms include human pathogens such as Vibrio cholera, Haemophilus influenza, Klebsiella oxytoca, and Campylobacter species. So we aim to understand how these mucosal colonizers interact with their host and how the host reacts towards the colonization. And in the case of the facultative pathogens, how can they persist outside of their host? So these aspects also will be the topic of our Life Science Tuesday on January 26th, and will now be introduced in more detail from my co-chair, Stefan Schild, together by introducing our speaker, Christoph Haug.
Okay, also a warm welcome from my side. As you can imagine from the title of our LST, we covered two interlinked research areas, infection biology and immunology. And infection biology basically focuses on the epidemiological and molecular principles of disease caused by bacteria, viruses, fungi, and parasites. And thus we welcome abstracts for our session covering any of these pathogens. Now, a lung infection, specific microbial effectors, also often referred to as virulence factors, enable microbial pathogens to not only successfully colonize the human host, but also to interfere in cellular processes and thereby causing the unique symptoms of the underlying disease. Importantly, evolution has not left us alone, but equipped us with a powerful army, the so-called immune system, which protects us from infections via various lines of defense. And this can roughly be divided into an acute response, which is the innate immune system, and in an adaptive response, which is the acquired immune system with a memory feature. Now, vice versa, microbes have acquired several countermeasures to bypass or disable our immune system. And thus, the pathogen host cell crosstalk is a vital warfare in our body, and with a battlefield of millions of deaths every year. Now, in summary, the research we are interested in our laboratories, and which will also be the main topics of our LST session, comprises the studies of infectious diseases, the identification of the causative agents and the microbial effectors, the downstream characterization of the pathogen-host interactions exemplified by immune modulatory processes, for example, as well as the crosstalk between pathogens and the microbes of our body, which can impact the microbial life cycle and physiology in our body. And overall, the elucidation of a molecular mechanism along these interactions may yield into new diagnostic or therapeutic strategies. And again, we cordially invite researchers to submit abstracts covering any of these aspects in our LST hosted in January. And this brings me finally to our invited speaker, which will be Christoph Haug, who is currently a professor for cell biology at the Department of Biology at the University of Constance in Germany. Christoph, along his research, is interested in the crosstalk between microbes and mucosal surfaces, especially how mucosal surfaces are protected from colonization by bacterial pathogens. And one of his model organisms is Neisseria gonorrhea, potent colonizer of the human urogenital tract, causing the sexually transmitted disease gonorrhea. A protection mechanism of our body against mucosal colonization is the so-called exfoliation, which is, generally speaking, a rapid shedding of our outermost epithelial cell surface layer to efficiently remove the attached bacteria. And what Christoph's work shows is that bacteria can counteract this exfoliation by binding to host cell receptors, which are called carcioembryonic antigen-related adhesion molecules, or also CCAMs, and by the release of a bacterial metabolite, nitric oxide. And we are looking forward to get more insights onto this pathogen host warfare along our LST. Last but not least, I also want to point out that our LST includes a webinar by Twist Biosciences with the title, "It's All About Design, Use of Next Generation DNA Libraries in Drug Discovery." So see you on January 26th.
Thank you, Sabine. Thank you, Stefan. I once more forgot to unmute myself. And I would ask Klaus Graumann and Georg Klima, are you ready to present?
I think we are.
Perfect. The stage is yours.
Great. Georg, will you start?
Yes, I will. Looks great. So thank you, you're all very warm welcome from my side to the introduction of the Life Science Tuesday number 7. It's called New Biologics and Novel Analytical Tools. It will take place on 12th of January 2021 already, so time is flying. My name is Georg Klima, and I'm currently heading Process Science at Boehringer Ingelheim, Vienna, and I will co-chair the session with Klaus Graumann, who's currently CEO at Fenestra, a startup company. And we are really glad that for the third time we can include talks about biotherapeutics development into the ÖGMBT Jahrestagung. And to kick off the session, we invited Jordan Turnbull. He's principal scientist at Viral Therapeutics, a company that works on oncolytic viruses. We think a very exciting modality to treat cancer. And Jordan will talk about the challenges of developing bioprocesses for those oncolytic viruses. And in particular, he will focus on product characterization, because the really product understanding will be key to implement the quality by design approach for new process developments. The work he will give insights in the development of novel purification techniques, complemented with extensive characterization using a broad range of analytical technologies. And with this brief introduction of the keynote speaker, I really would like to hand over to Klaus to introduce our session in more detail.
Thank you, Georg. As Georg already mentioned, this is already the third, only the third session, including biopharmaceutical industry topics in the Jahrestagung. And I think so far we had very interesting sessions, and we are also very hopeful that with this exciting topic about new therapeutic modalities, we can really collect a very interesting session again for this Life Science Tuesday. So as Georg already mentioned, for the keynote speaker, I think he will set the stage about these new drugs, which are very complex, of course. And we believe and we know that analytical tools especially will be needed to characterize and understand these products better, and especially also how they work in the human body eventually. So we are looking for contributions, especially for viruses, oncolytic viruses in this case, but in general, cell and gene therapies in the broader sense. But not only that, we also welcome contributions for other types of biopharmaceuticals, as always, relevant topics, hot topics that you may have on your plate. So we are very much looking forward to your submissions. Submission deadline is November 12th, and we will hopefully then, after November 12th, compile a very exciting overall program and share it with you, the complete agenda then soon afterwards. So thank you very much, and looking forward to a very exciting and interesting Life Science Tuesday, early January, excuse me.
Thank you for introducing your Life Science Tuesday in January. And we move on to Life Science Tuesday number 9, which will take place in February, in more detail, February 9. And the chairs of this Life Science Tuesday will be Beate Rinner and Cornelia Kasper.
Hello, can you hear me?
Yes, we can hear you, but the presentation is not yet started.
I can see the presentation, but I cannot hear my co-chair, Connie Kasper. Is she here?
She's muted.
Oh.
Ask her to unmute.
Hello?
Yeah.
Yes, we can hear you.
Hello. Sorry
No
for
problem.
the technical disturbance. My name is Cornelia Kasper, and I'm very happy to introduce the Life Science Tuesday number 9, which is called Tissue Engineering and Regenerative Medicine, together with my co-chair, Beate Rinner. And I would like to start by introducing our invited speaker, which is Professor Heike Walles. I've known her for several years. I don't tell you how many years. And I met her in Hanover, where we both started our early career. She was in a junior professorship at the medical school on tissue engineering. Then she moved forward to Stuttgart as head of the Institute for Cell Systems at the Fraunhofer Institute for Interfacial Engineering and Biotechnology. And after that, in 2009, she was appointed at the University in Würzburg at the first professorship in tissue engineering and regenerative medicine in Germany. And there she further developed a so-called Fraunhofer Translation Center in regenerative therapies. And since 2019, she's now professor at the Otto von Guericke University in Magdeburg as head of the core facility on tissue engineering. I'm very happy that she accepted our invitation, and her talk will be about the application of tissue engineering in the development of medicinal products. So it's a very interdisciplinary session, and the deadline for submission will be 9th of December. To introduce myself, just a few words. My name is Cornelia Kasper, as I told before, and I work mainly in the technological field in taking care about generating cells or isolating cells from patient donor tissue until we expand the cells and then do different things like differentiation, the classical tissue engineering approach, or where we also use maybe stem cells from donor tissue for the production of cell-based therapy products. The interesting point in my Institute of Cell and Tissue Culture Technologies is that we apply what we call physiological culture conditions, which include the application of hypoxia, so reduced oxygen concentrations in the cultivation containers. And we work in 3D cell culture systems under dynamic cultivation systems in so-called bioreactors. And we care a lot about the medium components, the composition of the medium, making sure that we work as far as possible xeno-free or maybe even chemically defined cell culture media. And I'm very happy also to just briefly let you know that I'm head of the working group on cell-based assays, therapies, and products. The working group was established in the ÖGMBT just recently, and we will place this announcement on the last slide. And I now hand over to my co-chair, Beate Renner.
Hello, can you hear me? Yes.
Yes, we can hear you.
Hello, my name is Beate Renner. I'm from the Medical University of Graz, from the Division of Biomedical Research, and I'm the head of the core facility Alternative Biomodels and Preclinical Imaging. Can you see the slide? Because I cannot see it. Ah, yes, thank you. Thank you very much.
You're not in presentation mode. That's the reason.
Pardon?
You have to start the presentation. We only have the edit view, not the presentation
Okay, but
view.
I cannot start it.
Small monitor at the symbol at the bottom.
I can handle that.
Yeah, now it's correct.
Thank you. Okay. I shortly want to introduce our core facility. We are divided into 3 parts. The first part is we provide cells for the medical university. That means we have the cell bank. It covers more than 170 cell lines. Then we offer a quality check, do cell-based assays and tumorigenicity assays. And then we would try to cover all the experiments from, not all experiments, but cell culture-based experiments from in vitro technology through preclinical imaging to in vivo technologies. We have preclinical imaging unit with a new, very and really great MicroCT. It's from Bruker, the SkyScan. Then we have a very innovative ultrasound system, and now we get a new optical imaging. Our aim is to really reduce animal stuff, animal experiments, and therefore we also collect all alternative models for animal experiments. And one of our key aim is to establish autologous cancer models. It means we have very rare cancer, for example, sarcoma or glioblastoma, where we have different types of tissue from one patient, and with that we isolate cancer stem cells and try to make cancer organoids and also co-cultures with cancer cells and also cancer-associated fibroblasts. Okay, then I would go further. Connie, could you please go further? Yes, and then I'm very happy and we're very lucky that we could get the speaker, Peter C. Johnson. He's a professor at the University of Southampton. He's also a supervisor in a supervisor board of several companies. And if we spoke in modern language, we can say he's just an influencer because he's a specialist in the field of tissue engineering and stem cells research. And we are very proud that we have him as a special lecturer. But we also are very lucky if you send us some abstracts, and the deadline for submission of our session is the 9th of December. Thank you very much.
Thank you for presenting Life Science Tuesday number 9. And with this, we move on to Life Science Tuesday number 11, which is presented by Michael Bergmann and Gerald Höfler. Today, just by Michael Bergmann.
Okay, thank you. Can you see the presentation?
Yes, looks
yes.
Good.
Yes,
audio presentation is fine.
So our Tuesday is about translational oncology. It's on March 9. And the invited talk will be by Daniela Tommen from the Netherlands Cancer Institute. And it will be chaired by Gerald Höfler and myself. Wait a minute, how do I go on with the slides? Oh, okay. So let me just introduce the field of translational oncology. So clinical oncology is basically that there is a tumor which we might treat by chemo. The tumor gets smaller, we might do chemo, and then again there is some disease-free and overall survival. So translational oncology is really that is a new field, and it's a difficult field because it should bring together the PhD fields together with the MD field, and as you know, there is a big gap. So what translational tries to close this gap. And why is this so important? Because if we treat patients in our scheme, we often treat patients which do not respond to our therapy, and so the molecular biology supposedly should help us that we have predictive and predictive parameters which just indicate which therapy is really working, and also possibly prognostic parameters which things how is the overall survival, and understanding the disease is really providing us novel therapies. And so in order to do that, it's really important that you on one hand understand the clinical field, but also understanding the molecular biology methods, which are just made sort of for quantitative immunohistochemistry, but it could be also FACS analysis or whatever. So does this work? Yes, it really does work. And one success story in translational medicine is understanding the tumor microenvironment of human cancer. And I think Jérôme Galland was really one of the first ones who really sort of brought more attention to that, because a tumor does not only consist of tumor cells, but also of immune cells. And importantly, whether you have a lot of the T cells, it's CD3 or CD45 cells. So Jérôme really made a score based on quantitative immunohistochemistry, and he was actually a bioinformatician. That really changes your prognosis in human cancer. And this in this prognostic factor of this tumor immunology is more relevant than any other prognostic factors we had to date. So that also brings us a better understanding of the mechanism of the disease, because if we can target those T cells to be sort of more reactive or better within the tumor, we potentially have a better cure for cancer. And whether this is true, you know that for the Nobel Prize winner, so there is on those T cells, there are checkpoints, because the tumors really inhibit T cells that the T cells can sort of fight the tumors. And if you now inhibit those inhibitory receptors by sort of blockers of the PD-1, PD-L1 interactions or CTLA-4 interactions, then those T cells are again reactive, and they will cure cancer, which you can't cure otherwise. And what you can see in this plot, this is, for example, such a inhibitor of checkpoint inhibitor for non-small cell lung cancer in stage 3, and you see they have sort of a prolonged survival rate, and as compared to chemotherapy, where the curve really went down after a certain time, and there was just sort of a little bit of an increase of the 50% disease-free survival. This really stayed long, and this comes here comes in our keynote speaker, which is Daniela Tommen, because of course, if you have such immunotherapy, you don't know which patients really respond and which don't respond. And she made important contributions. So she found in this non-small cell lung cancer that those tumors which have T cells which very high levels of the PD-L1, and this must be more than 1% of those T cells, as you can see in this right side of the slide, then this really responds. So really to find predictive factors is very important, and I think Daniela Tommen made here an important contribution. So what she also was, she also thought of the best sort of an ideal clinical setting, also bringing together those both worlds of the PhD world and of the MD world. So the clinical, so she thought of what's the best study design. So then classical study design is that you have any cancer, and then you try different therapies, as for example depicted here. But obviously, if you do that, there is an enormous number of patients which are sort of not well treated, which would respond sort of the blue one. Possible patients would be in this cohort, but they better respond to this cohort. So another method of personalized cancer immunotherapy will be that if you do sort of your gene expression analysis or any other analysis, and then based on those predictive markers, then you give therapies, and that might be already sort of a little bit sort of a more rational design. And a further studies design, which she really promoted, is that you start with, in this case, neoadjuvant trial, which means to give the chemotherapy before the surgery, and then you have responders and non-responders. So the responders is fine, and you keep treating that. But if you have the non-responders, you take small pieces of the tumor, you make a short-term tumor culture, and then you check some of your other drugs or drug combinations, and you try this one. Again, you have some responders, which are fine now, and some non-responders, where you do the next thing. So what you do in this trial concept, which she called the Lombard Street, according to the San Francisco road, this Lombard Street. So basically, you ideally combine the clinical need of one cohort of patients with personalized short-term cultures. So what about the person of Daniela Thormann? She did a PhD in T-cell immunology, but after she studied actually medicine, and she did a residency in internal medicine in Switzerland. She did training in internal medicine and in oncology, and in parallel, she did a postdoc in a cancer immunology lab in Switzerland. Then she joined Stan Schumacher lab. That's why she got involved in this heterogeneity of T cells and the outcome of tumor immunity, and publishing this Nature Medicine paper. And since 2020, she's a principal investigator at the National Cancer Institute in Amsterdam in Holland. And her main question is really establishing primary ex vivo cultures, investigating immunomodulatory drug combinations. So again, translational oncology is what I told you. We try to molecularly understand cancer, to have predictive marker, prognostic markers, and novel therapies. And I think our keynote speaker ideally does this. Now this brings me to the chairs. So there is Gerald Höfler, who couldn't make it today, but most of you might know him. He's professor of pathology and the head of the Institute of Pathology in Graz. I mean, he has a sort of an age index of 52. So he is currently working on lipid metabolism. So he's interested how the adipose glycerate lipase plays a crucial role in the development of tumor-associated cachexia. And in addition, whether the loss of adipose triglyceride lipase is associated with cancer development. Myself, I'm sort of similar working in the field of Daniela Thormann. I initially sort of developed the first oncolytic flu virus. I became very interested in the tumor microenvironment. I have some publications about markers, specifically in colorectal cancers. We have now studies where we use neoadjuvant checkpoints for rectal cancers, and we do others for GI cancers if we get funded by IIT trials. And I also established now a collaboration, so I have a collaboration with Boehringer Ingelheim, where we also do those short-term cultures. These are sort of 2-3 days cultures where we check out novel immune modulatory drugs. So I hope you will join our session. Again, it's on March 9, 2 months before. So this is January 9. There is the deadline for the abstracts. And we have a webinar for single cell multiomics and spatial gene expression analysis by 10x Genomics. And as you could see from our slides, we believe that immunohistochemistry gives you an enormous wealth of data. And I think it's important to understand disease and guide medical therapy, including surgery and oncology. Thank you very much.
Thank
So
you.
I have to stop that.
Sorry.
Yes, please, on the top, yeah. Thank you for giving this overview on your Life Science Tuesday. And with this, we move on to the final Life Science Tuesday of this series. And to my knowledge, Bernd Niedetzky will not present today. It will be only Robert Kurist. Stage is yours.
Yeah, thank you, Harald. Give me a minute to activate the presentation, please. So, yeah, thanks also from my side to the organizers to make this happen, and in this conference-free time provide such an interesting program, and also for all your efforts in this many preparatory meetings we have been invited so far. So this is a bit of change of topic. This session is about this is the last session, and this is industrial biotechnology. It's called Production System of the Future. So methodologically, it means bioproduction, industrial biotechnology, fermentation, biocatalysis. And the topic is also a bit molecular biotechnology meets bioprocess engineering. And these are the two chairs. So this is me. I'm professor for molecular biotechnology at TU Graz. And Bernd, who can't be here today, is professor for bioprocessing at TU Graz. And my research is biocatalysis, enzyme engineering, and recently we've been also trying to use autotrophic microorganisms as catalysts, and this means to use hydrogen or sunlight to drive biocatalysis, which is an exciting concept, I think, but also a bit far from application, and this brings us to the topic of the future production systems. This is a bit the problem our society has right now, which is where will the future resources come from. So on the left side, you see there are fossil resources. These are established routes, but this creates a lot of waste and consumes a lot of energy. And we also do not know if this resource base will hold in the future. On the right-hand side, we see agricultural products, and there the volumes are limited, and we also compete with the use of for food. So it will be very difficult here to deviate biomass for the production of chemicals. And of course, utilization of side streams could be interesting, but this is also a very limited volume. I think, for instance, a country like Germany has less than 1 million tons of straw per year. And in the center, we see new production systems. This could be autotrophic microorganisms, cyanobacteria, carbon monoxide using organisms, hydrogen utilizing organisms. So there's an exciting diversity of metabolism. And I think it's quite clear that all three columns will contribute to our resources in the future. On the left side, future production systems means to make environmental friendly catalysts and environmental friendly cells. So here we would like to invite abstracts and contributions. On the right side, this is bio-based renewable resources. This here also means to integrate technology. Here the challenge will be to integrate molecular engineering and bioprocess engineering. And in the center, this is a technology which is very far from application, but could radically improve the situation. And here I'm very happy to present Stéphane Gouillet. He is from Toulouse, and he is working on hydrogen utilizing bacteria. And the title here, Metabolic Pathway and Bioreactor Engineering for Biofuel and Chemical Production from Carbon Dioxide and Cucurbitacus necator. Here the title is really program, because what distinguishes Stéphane's research is that he integrates bioprocess engineering with molecular techniques. And I think this is the strategy to be successful with new production organisms. For instance, if you want to drive a bacterium with hydrogen or with carbon monoxide from a steel mill, then the first problem is how to dissolve the gas in water. This is very difficult. How to reach good cell densities with hydrogen utilization. There's a problem. It's an aerobic process. So this would be explosive gas mixtures. And Stéphane really has made fantastic contributions that actually the cultivations work reliably. And here it really has the dimension to work on the molecular dimension, on the molecular level, and at the same time to verify the systems in a process environment. So I hope this is will be a very exciting talk. Abstract submission will be possible until January 23. And this will be just one topic here, the hydrogen utilization. And I would like to encourage all who work on biocatalysis, exciting cell production systems to contribute. And I'm looking very much forward to the session, and thank again the organizers here for making this kickoff happen. Thank you.
Thank you, Robert, for presenting the final Life Science Tuesday of this series. And with this, I would like to share some more information with you. And thank you to all of the chairs who presented today. We know it's, in organizational terms, quite a challenge to go virtual. We learn that day by day. Thank you for investing so much time. Then one more piece of information. For each and every Life Science Tuesday, there is a short video available on YouTube, which you may, as chairs and we as organizers of this conference, also use to promote the Life Science Tuesdays further. And now I think we do still have a few minutes to have a short breakout session for the Life Science Tuesdays number 7 to number 12. Okay, so let's share this format. So let's wrap up what we have shown so far and discussed so far. So we're at the end of my Life Science Tuesday event and session, and also the kickoff of the virtual expo. We are looking forward to one more day of, in the morning, a scientific session on healthy aging, and in the afternoon, a similar format to what we had today, but in this case, on career opportunities in life sciences, and also the kickoff of the virtual Life Science Career Fair. So as you will understand, we see these kickoff days, as the name says, as kickoff of several platforms for interaction, which we wanted to introduce. On the one hand side, the scientific sessions, the Life Science Tuesdays. On the other hand, the virtual expo and virtual Life Science Career Fair. And as promised yesterday in the afternoon, we add more and more interaction possibilities that you will hopefully use quite a bit over the next few months. And with this, I would like to switch from Life Science Tuesday announcements to thanking the sponsors that we have for this conference. And finally, ultimately, I would, if there's need, also hand over back to Alexandra if I've forgotten anything for today. If there's nothing, she's shaking her head. I thank you for your participation, for your contributions, for your interest and patience with the little technical issues that we still have. But it's a learning phase, and we'll learn for future Life Science Tuesdays. And when we are perfect in doing all that, we hope that we will have face-to-face meetings once more at this moment. So with that, I thank you, and looking forward to seeing you again tomorrow.
Okay. I would also like to thank all of you, all the chairs and all the participants, and also the companies that are here and presented their web pages. And I'm also looking forward to see you tomorrow. Okay. Bye.
Thank you. Bye.